Endospores are produced by a limited number of organisms. There's only a few. And the only two that you need to be concerned with in here are Bacillus species and Clostridium species. Both of those are known sporulators. Bacillus primarily causes a lot of problems with food preservation. So home canned green beans or food spoilage can occur with Bacillus, but you do have a few that are famous, like anthrax. Bacillus anthracis can cause major problems. Clostridium has four main pathogens. They're Clostridium botulinum, botulinum toxins. Newborn babies should never get honey because of botulinum toxicity. The spores are found in the saliva of the honeybee. So, since young babies don't start producing acid until in their stomachs until they're about 18 months of any significance, then it doesn't inhibit the spores from forming. So you get what's known as limp neck or flaccid neck syndrome in babies. They are given that. So you've got botulinum, gas gangrene, which is Clostridium perfringens, tetanus, Clostridium tetani, and Clostridium difficile are the four main organisms in the Clostridium group. The difference between the Bacillus and the Clostridium primarily is that Bacillus is aerobic, Clostridium is anaerobic. They're able to form these resting endospore cells, which are really hard to get rid of. It's one of the reasons that we have to autoclave our media, is because the spores are pretty much everywhere. And there are high numbers of them in soil. You find them almost ubiquitously in every lab or in every hospital room. They're highly durable. They can set in, well, in England they put a bunch of them in little vacuum tubes and sealed them up over 100 years ago. And every 10 years they break one open and they culture it. And they're still growing. They've also isolated spores from the guts of honeybees that have been embedded in amber for 10,000 years or some crazy number like that. And they grew. They grew from as aseptically as they could take those things out. That just is amazing to me. There's some that you can boil in boiling water for 12 hours, and then if you put them in the right environment, they just germinate right on. They're highly resistant to chemicals, UV light, radiation, and the resistance is primarily due to these 7 or layers of calcium dipicolinic acid or calcium dipicolinate that wraps around a protein coat of the spore. It's a spore coat, and it's wrapped up in that calcium dipicolinic acid, and it makes up about 15% of the dry weight of the spore, but it contributes strongly to the survival of the spores. If you were to grow sporulating organisms in calcium-free solutions, then the spores that they produce are not nearly as resistant as those that have the calcium present. Okay, long-term survival, hundreds of years, isolated from guts of insects. There's just some really interesting topics that talk about endospores. I'm going to have Mr. Watkins do a video on this because he is the spore expert. He knows a lot more about it than I do, but this is a basic overview of endospores.